What are MCAT Bio Biochem pH and buffering basics? Have you ever wondered how our bodies keep everything running smoothly, even when faced with changes in our environment? One of the key players in this process is pH and buffering. Understanding these concepts is essential for your medical college admission test preparation, especially in the biology and biochemistry sections. Let's break it down in a way that makes it easy to grasp. First, pH is a measure of how acidic or basic a solution is. It tells us about the concentration of hydrogen ions in a solution. The formula for calculating pH is the negative logarithm of the hydrogen ion concentration. So, when you see a lower pH, it means the solution is more acidic. Conversely, a higher pH indicates a more basic solution. For example, human blood typically maintains a pH around 7.4, which is slightly basic. Now, let's talk about buffers. A buffer is a solution that helps resist changes in pH when small amounts of acid or base are added. Buffers usually consist of a weak acid and its conjugate base or a weak base and its conjugate acid. They work by neutralizing added acids or bases through reversible reactions. This means they can stabilize the pH, keeping it within a narrow range that is vital for cellular function. When an acid is added to a buffer, the conjugate base reacts with the excess hydrogen ions. This reaction forms the weak acid, preventing a significant change in pH on the flip side. If a base is added, the weak acid donates hydrogen ions to neutralize the hydroxide ions, again minimizing any pH shifts. A handy tool for understanding how buffers work is the Henderson-Hasselbalch equation. This equation connects the pH of a buffer solution to the acid dissociation constant and the ratio of the concentrations of the conjugate base and acid. When the ratio of the conjugate base to acid is close to 1:1, the buffering capacity is at its best, and the pH is approximately equal to the acid dissociation constant. In our bodies, one of the most important buffering systems is the carbonic acid and bicarbonate ion system. This system keeps blood pH around 7.4, which is crucial for maintaining proper cellular activities. If blood strays too far from this range, it can disrupt many vital processes. For your MCAT preparation, here are some practical tips. Make sure to memorize strong acids, which fully dissociate, and weak acids and bases, which only partially dissociate. Practice applying the Henderson-Hasselbalch equation quickly, especially since you won't have a calculator during the test. Familiarize yourself with buffer systems in physiological contexts, like blood, and how they respond to changes in acidity or basicity. Lastly, understanding titration curves can be beneficial. These curves show how changes when strong acids or bases are added to weak acids or bases, highlighting the regions where buffers work best. By mastering these concepts, you will build a solid foundation for your medical school journey and succeed on the medical college admission test.